Hello everyone, and thanks for joining today's webinar. I'm Minkyung Baek from the Department of Biological Science at the Seoul National University. So today we're discussing how AI has revolutionized our ability to predict protein structures and interactions, and also design new functional proteins, mainly focusing on advances my colleagues and I have made in this rapidly evolving field. So, as you know, last year, the Nobel Prize in Chemistry was awarded for the groundbreaking advancement in the field of protein structure prediction and also protein design. And this achievement highlights the huge impact of computational tools like AlphaFold and RoseTTAFold on modern biology. So, as someone fortunate to have been mentored by David Baker, one of the pioneers in this field, and also my postdoc advisor, I had a chance to visit Stockholm during the Nobel Week and meet many Nobel laureates in person. So the topic of my presentation today is closely connected to this Nobel winning field. And in today's talk, I will discuss AI-based approaches for protein structure prediction and protein interaction prediction, and also protein design. So let's start with a quick reminder of the central dogma of molecular biology. So as we know, the DNA is transcribed into mRNA, which is then translated into proteins, the basically workhorses of our cells. And proteins perform an incredible array of functions in living organisms. So they basically facilitate transport, act as enzymes, catalyzing various biological reactions. They also participate in signal transduction pathways and form essential component of our immune system. And the function of protein is actually tied to its three-dimensional structure. And this three-dimensional structure emerges as amino acid chains fold into the complex shapes, driven by various chemical interactions. And for decades, determining those protein structures experimentally has been very challenging and also time-consuming and also expensive, which creates a bottleneck in our understanding of protein function. So in those regions, while the number of protein sequences is rapidly growing, the number of experimentally determined structure is slowly increasing. So in this figure in 2022, we only have 195,000 structures. Now we have like 240,000 structures in the protein data bank. Then how can you fill this gap? How can you get 3D structures of proteins that can help us understand their function? So to fill this gap computationally, many researchers have been worked to develop protein structure prediction method that basically predict the 3D structures from each amino acid sequence, and also they explored various ideas. And I want to share one of the key concepts that really changed how we predict the protein structures. So we know that protein shapes determines its function. So if the structure changes too much, the protein usually can't do their job anymore. So take a look at this myoglobin example. And in this figure, it shows the myoglobin from 5 different animals. And those animals evolved separately for millions of years. But you can notice something interesting. So basically, their myoglobin structures look almost identical. And this is because they all need myoglobin to bind oxygen efficiently so that it can supply oxygen to their muscle tissue. So during the evolution, nature is quite practical. So when random mutations happen, those that mess up the protein's function get eliminated. But mutations that keep the structure working properly can stick around. And this is why functional structures tend to be preserved over time, as you can see in this example. So this leads us to really useful insight. So proteins that share a common ancestor, what we call homologs, tend to maintain similar structures even as their sequences become quite different. So we can use this principle by collecting the related sequences and aligning them into what we call the multiple sequence alignment in short MSAs. And those MSAs deliver important structural information. So when we see certain positions in the protein changing together across species, it usually means these positions are physically connected in the 3D structure. So for example, if two amino acids need to fit together in the protein core, and one changes to bigger amino acid, then the other might change to a smaller one to make a space. So basically, they compensate for each other. So basically, we can find some structural patterns from the MSA, something like those coevolution patterns. Then the question is, how can we efficiently extract those structural patterns from the MSA? And this is where AI come into play. As illustrated here, AI algorithms attempt to learn multiple levels of representation using a hierarchy of layers. And they're remarkably effective at identifying some patterns, basically what we need for protein structure prediction. So for protein structure prediction, we need to detect those structural patterns hidden in the multiple sequence alignment. And when we provide the sufficient training examples, those AI systems begin to recognize those hidden patterns in the data and respond in useful way. So in protein structure prediction problem, if AI can learn those relationships between sequence patterns in the MSA and the resulting 3D structures, then we can predict 3D structures precisely just based on the MSA information. So our approach for protein structure prediction, named as RoseTTAFold, and this basically has a three-track attention-based network. And we first published the original paper in Science in 2021, and the RoseTTAFold 2 preprint on a bioarchive. And in this model, we basically try to make the AI model that can encode those structural patterns hidden in MSA to predict the 3D structures of our carrier protein. And the RoseTTAFold basically processes those information along these three parallel tracks. The first 1D track that handles sequence information from the MSA so that it can understand the sequence alignment and tries to extract the useful residue, the contact information from there. And the 2D track that basically captures the pairwise relationship between the residues, basically a structured patterns encoded in the MSA. And the third 3D track that basically builds and refines the actual 3D structure. And those three tracks communicate with each other through the attention mechanisms, allowing information to flow between sequence, pairwise interactions, and 3D representations. And this network iteratively refines its predictions through the multiple cycles and achieving progressively more accurate structure. And the effectiveness of AI-based approaches became dramatically apparent in the 14th critical assessment of protein structure prediction, the Cas14 experiment happened in 2020. So as shown in this graph, AlphaFold 2 from DeepMind achieved the high accuracy with an average GDT score close to 90, which is considered roughly equivalent to the experimental determined structures. And our RoseTTAFold model also performed remarkably well, and the latest model shows the almost same performance. And this actually represented a genuine paradigm shift in structural biology. So for the first time, we had some computational method capable of predicting protein structures with accuracy approaching the experimental method, but at the fraction of the time and cost. And those examples on the right illustrate the quality of the predictions. The green structures are experimental result, and the blue ones are computational predictions. And as you can see, their agreement is pretty remarkable. So with AI, we can predict the protein structures in high accuracy. But protein structure prediction is not the end of the story. What we want is basically understand protein function by understanding their interactions. So for example, let me ask you whether you can guess the function of this green protein just based on the structure. This green protein is overexpressed in cancer cells, and we can predict or determine the 3D structures using AI or experimental method. And even though we know their 3D structure, it's very hard to say why this protein is related to the cancer. But what if we know the interaction partner of this green protein? So if you discover this green protein binds to the PD-1 protein on immune cells, colored pink here, then you may conclude that it helps the cancer cells evade the immune system by suppressing the immune cell, basically the T cell activity. So all the examples I showed earlier by mentioning about their protein's function and their relationship to the protein structure, actually proteins do their functions by interacting with other biomolecules. So what we need to do is basically predicting not only protein structures, but also their interactions to understand their function. And just like with individual proteins, actually evolution leaves some kind of signatures on interacting protein pairs. And here shows how. So when two proteins physically interact, certain amino acids on their surface often need to maintain compatible physicochemical properties, such as charge, shape, and hydrophobicity, so that those binding interfaces remain stable over evolutionary time. So as a result, if a residue in one protein mutates, then the interacting residue in each partner protein may also mutate in a compensatory way, which called as a coevolution. And those correlated changes can be detected by comparing the sequences of interacting protein pairs across many species, as similar in the monomeric protein structure prediction. So if we can align orthologs of protein A and B from dozens or hundreds of species, then we can look for patterns of statistical coupling between residues. And strong signals strong signals means those proteins interact in a conserved manner. So in other words, the evolutionary pressure not only preserves individual protein structures, but also preserves their interactions. So by analyzing those paradigm assays, we can extract information about whether two proteins are likely to interact, and even which residues are likely to be in contact at the interface and form which kind of complex structure. And this is the same core idea that powered the monomeric protein structure prediction, just it extended to the level of the interprotein contact. And that's why we can still utilize the RoseTTAFold and AlphaFold for the PPI prediction. So to explore protein structure prediction AI's potential for the PPI prediction, we developed a pipeline to predict protein-protein interactions across entire proteomes. And we tested our ideas on the yeast interactome. Basically, we screened the tens of thousands of protein pairs to detect the reasonable protein-protein interactions. So we generated a bunch of paradigm assays for all possible protein-protein pairs in the yeast and passed them through the RoseTTAFold and AlphaFold and predicted their interactions as well as their complex structures. And these allowed us to generate a large set of high-confidence interaction models, many of which were later validated experimentally. And it was one of the first large-scale demonstrations that AI can be used for protein-wide structure-based interaction mapping. And in our East PPI prediction project, one of the bottleneck was computational efficiency. The RoseTTAFold was fast, but less accurate than AlphaFold 2. And AlphaFold 2 was accurate, but computationally expensive. So we developed the RoseTTAFold 2 Lite, in short RF Lite, basically an optimized version of the RoseTTAFold, specifically designed for faster and more accurate PPI screening. So our previous screening pipeline used for East interaction screening basically employed a two-stage approach. First, screening using the RoseTTAFold 2 track, which is fast but less precise. Then do a AlphaFold 2 screening, which is somewhat slow but more precise. And we hope by developing this RF2 Lite improve the RoseTTAFold 2 track by incorporating some concepts from the AlphaFold 2, such as recycling, self-distillation, and also use of proper inductive bias, something like pair former. And also, we also hope it's more computationally efficient than AlphaFold 2, so that we can still do a fast first screening, but with improved precision. So we basically achieved this by reducing the computational complexity from L cubic to L square through the structure-guided pair attention